Our goal that we chose from the 17 Goals of Sustainable Development was Goal 15, Life on Land. Our target specifically talks about reducing the degradation of natural habitats, halting the loss of biodiversity, and protecting and preventing the extinction of a threatened species. Throughout our presentation, we talk about how you can use gene modifiers and editors such as CRISPR-Cas9 and DNA barcoding in order to reduce the degradation of natural habitats, halt the loss of biodiversity, and protect and prevent the extinction of a threatened species. We specifically go into detail about the Tasmanian devil, a current endangered species located in Tasmania, as an example for how we would use this.
Biodiversity. How modern science, particularly CRISPR-Cas9, can assist in preserving species like the Tasmanian devil facing extinction. Biodiversity is essential for maintaining a healthy balance in our ecosystems. The Tasmanian devil, a unique species, is endangered due to devil facial tumor disease, a highly contagious cancer transmitted through biting during mating. The low genetic variance in Tasmanian devil populations make them susceptible to DFTD. This disease likely originates from a single Schwann cell, causing an 80% decline in their population in just 2 decades. CRISPR-Cas9 is a game changer in genetics. It uses RNA to guide an enzyme, Cas9, to specific DNA sequences, allowing precise genetic modifications. CRISPR offers hope for conservation by helping species adapt to new environments, resist invasive species, and combat diseases. CRISPR enables targeted genetic modification through homology-directed repair. This precision can boost genetic diversity in endangered species like the Tasmanian devil. Before using CRISPR and gene editing to address biodiversity issues, we must think about the ethical concerns. We may not fully understand the consequences which could harm animals' health with unexpected problems. CRISPR-Cas9 could increase the genetic diversity of Tasmanian devils, making them resistant to DFTD. Even small genetic changes could enhance natural selection, reducing disease-related deaths over time. In conclusion, CRISPR-Cas9 offers hope for preserving species on the verge of extinction.
Deforestation and natural habitats. Australia is the only nation in the developed world to make the World Wildlife Fund's global list of deforestation hotspot. The Benign Living Planet report published puts Australia alongside New Guinea, Indonesia, Congo and Brazil for deforestation. Walking through the forest, I was wondering about the reasons. The main cause of this is land clearing for livestock, according to Dr. Martin Taylor, and Queensland is on the top of the list. In 2015-16, about 3,95,000 hectares of native vegetation were cleared. It's equal to destroy 1,500 football fields daily. The simplest action in this case can be Newton's third law. Where a person acts, the other should react. If we achieve such an equilibrium, much of the problem would be solved. But it needs to be scientific, just like the law. And while replanting, we can use genetics, gene-modified plants that are more reluctant to pests and have rapid growth response, while restoring forests and fixing habitats. DNA barcoding can help in species identification. Metagenomics and biomolecular techniques can be used to study the microbial communities and to know about the diversity among organisms. It would be helpful in choosing more friendly species to make long-lasting environments. This biomolecular data can be integrated with other environmental data to get a more comprehensive image of deforestation. It can be used to make more fruitful policies.
Gene editing and modifying may be a promising solution to these issues if we can comprehensively understand our ethical implications.